Hello, everyone, and welcome back to genetics class. We are going to be doing a bunch of fun stuff today, but some of it might be a little confusing, as always. So I hope you listen carefully, and I hope I can make it clear for you. Let's get started. On today's schedule, we're going to be finishing up meiosis from the last lecture. I'm going to clear up some terminology that I have not been defining and maybe a little confusing to people. And then we're going to get into the meat of this lecture, chromosome packing, sex-linked traits again, non-disjunction, and then we're going to spend a lot of time on probability, combinatorics, and chi-squared. And I actually doubt we're going to get through the whole thing today. That last topic is going to be probably the hardest out of everything we're covering today, and I'm guessing about half of it is going to get shunted into lecture 6. But we'll see. Let's get started with finishing up meiosis. We left off, we covered meiosis in humans and animals in general, but meiosis proceeds a little bit differently in plants. Well, let's clarify that. It's not so much that meiosis is different, it's just that what meiosis is used for is different. If you have heard of alteration of generations, then that's really what I'm getting at here. Humans have essentially two aspects of our generation. We have the diploid stage and the haploid stage. And for us, the diploid stage is almost the entirety of life. The haploid stage is not independent at any point. So the haploids in humans are just the sperm and the egg. And they really don't spend any time unsupervised. They are always either internal to our bodies or are in the process of fertilization, which again happens internally. So plants and algae, some algae, they actually have their haploid stages external. And I don't mean like in frogs where it's external fertilization, where, the female frog lays a bunch of eggs and the male frog spits out its sperm into the water and they get fertilized that way. That's not what I'm talking about, because that's still just the gametes, the haploid stage solely exists for the purpose of fertilization to get right back into the diploid stage. In plants and algae, the haploid stage is entirely a full-fledged life state. You can think of it almost as the sperm and the egg of humans going out and living a full-fledged, fulfilling 80-year-long life before undergoing mitosis to then fuse into humans. And I'll get into this in a minute. But okay, strictly speaking, humans are diploid gametophytes. A gametophyte is an organism that creates gametes. That's what it means. We create gametes. Then there is also the sporophyte, and that is an organism that develops spores. Or at least a life stage that develops spores. So we are gametophytes, we produce gametes, and then the gametes fuse to produce more gametophytes. And while we are gametophytes, we are diploid. We have 2 copies of every chromosome. And then our gametes are haploid. They have 1 copy of every chromosome, right? That's review. We've covered this already. The way it works in plants is the gametes fuse by fertilization, and they produce a diploid organism. But that diploid organism is a sporophyte, because when that undergoes meiosis, it does not create gametes. It is not a gametophyte, a thing that creates gametes. It is a sporophyte, a thing that creates spores. I'm realizing I probably should have included an image of this in the slide or in a slide of its own, and I did not think to. Please, by all means, look this up. Look at images online. It is a cycle. Sporophytes undergo meiosis, and they produce haploid cells that are spores. Those haploid spores undergo mitosis. So they will take their individual chromatids, they will duplicate them, and then segregate those chromatids into separate cells. So it goes from a single haploid cell into two haploid cells using normal mitosis, like we covered. Our mitosis that we talked about was diploid mitosis, where the pairs of sister chromatids got separated. This is a diploid mitosis. And what the spores create when they undergo this mitosis and become a multicellular thing, that multicellular thing is the gametophyte. That is a stage of life that lives out on its own in the world and eventually creates gametes. Now, when we create gametes, we are diploid and our gametes have to be haploid, so we undergo meiosis to decrease the number of chromosomes, or at least decrease the numbers of copies of chromosomes. The gametes in plants are already, or I'm sorry, the gametophytes in plants are already haploid. So they don't need to do a meiosis to become haploid. They just undergo more mitosis and create gametes. And then these gametes fuse through fertilization, and the gametes can be different individuals in the same species, or they can be different cells from the same individual. But they fuse to form a diploid. That's fertilization. And that diploid grows up into a sporophyte. So in humans, we go from gametophyte, gamete, gametophyte, gamete. And the gamete stage of our life is minuscule. Whereas these go from gamete to sporophyte, to spore to gametophyte, to gamete to sporophyte to spore to gametophyte, and so on. So they have 4 stages of their life while we have 2. This is considered alteration of generations because one generation being the sporophyte, the other generation being the gametophyte, and it alternates back and forth and back and forth. I don't think that's a good name because, as far as I'm concerned, it's a cycle of generations, not an alternation, or a cycle of stages. Now, do you know any gametophytes? Are you familiar with any? Let's find out. If you are familiar with the sort of soft green carpet-like moss, sphagnum moss, that sort of covers natural wooded areas, that is the gametophyte form of that particular species. And the sporophyte form are the tiny little flower-like things that come up out of it. Pine nuts are the gametophyte stage of pine trees. And ferns often have a little heart-shaped leaf that you can see, as opposed to the large fronds. And the little heart-shaped leaves are the gametophyte stage of ferns. So that's a very brief rundown of alteration of generations and how meiosis is used for different purposes in plants. Now I want to talk about some terms that may be a little confusing to you that I've been using, or at least the differences between the terms might be confusing. So let's get into that. The biggest confusion that I tend to see in genetics classes is people using these 4 or 5 terms interchangeably: gene, allele, gamete, locus, and loci. They are not interchangeable. They are very different from each other. And the biggest confusion here is between gene and allele. So I want to clear that up right now. When I talk about a gene, when geneticists or biologists talk about genes, they are talking about a region of DNA on a chromosome that encodes a protein. And we're specific about which gene we're talking about in general. The thing about a gene is, in any given species, a gene is a nebulous thing. There is no one correct DNA sequence for that gene. There is no one thing that we can say, this is the, this is the Sonic Hedgehog gene. You may think that was a random example, but I'm going to explain it in just a minute. The reason that it is nebulous and not specific is because if you look at the 8 billion humans on this planet, you might, and if you look at the sequence of each 8 billion people's Sonic Hedgehog genes, you might get 6 billion different gene sequences. It's not one identical sequence because everybody's got like one point mutation here, a different point mutation there, and everyone's individual specific point mutations more or less are independent and different from each other. There are constantly mutations arising in our DNA through every single mitosis, and there are mutations back to what a previous sequence had been, and so on. As long as the protein that is made from that gene stays functional and that individual survives to breed, then that particular sequence of that gene stays in the population. So the exact sequence is different for most people or large quantities of people. But when you look at all 8 billion people and you compare all of their gene sequences for any one given gene, 90% of the people all have the same base at position 1. 90% of the people all have the same base at position 2. And that 90% could be a different 90% than base 1. And 90% of the people have the same base at position 3. And that could be a different 90% of people. So, in general, we all have more or less the same sequence for any given gene. But there's spots here and there where you might be different, and then there's a spot over here where he might be different, and a spot over there where she might be different. But on the whole, it's all the same. Now, each gene is only present once in your chromosomes. Well, it's only present once in your haploid chromosomes. In your normal everyday diploid cells, you have one copy of that gene from mom and one copy of that gene from dad. Now, throughout the course of evolutionary history, our genomes have duplicated, individual genes in our genomes have duplicated, and so on and so forth, and that has risen to multiple copies of given genes. But here's the thing about that. Over evolutionary time, those multiple copies of a given gene mutate independently of each other. And so after a while, they become distinct from each other. They become different genes. And they wind up having slightly modified functional capabilities. They might be expressed in different cells and at different times. And so really, they become independent of each other. Closely related, but independent. And so they are considered different genes. And one frequently studied example of multiple members of a given gene family are the hedgehog genes. Now, Drosophila, the fruit fly, has not undergone quite as many gene duplications or genome duplications as humans have. So a lot of the genes that we have in multiple copies, they only have in one copy. And this is the case for a gene called hedgehog. And in Drosophila, mutants, or genes rather, are named after what the mutation of that gene looks like. So in the early 1980s, a pair of Drosophila geneticists created hundreds of mutations in Drosophila and then started characterizing each of them and identifying where on the chromosomes each of these mutations arose, and then identifying the gene sequences that they came from. And they named them all after what they looked like. And that's where things like hunchback and giant and bicoid and hedgehog all come from. Well, about 10 years later, in the early 1990s, um, some scientists in Boston were studying, hey, we know that the fruit fly has a hedgehog gene, and we know that a lot of the genes in flies are shared in other animals, so let's see if humans have a hedgehog gene. And this is before DNA sequencing was all that easy, so there's other methods that they used to find out whether the gene existed. And when you go from one organism to another, you can rename a gene, but vary it on a theme. And so the scientist who discovered the first vertebrate, the first vertebrate hedgehog gene, he named it Sonic Hedgehog because this was the early 90s and the Sega game Sonic the Hedgehog was very new and very popular. So he named it after that. I know this because I worked for the guy for about a year and a half, and he told me himself. The reason I'm telling you about Hedgehog, or Sonic Hedgehog, is because once DNA techniques became a little bit more sophisticated throughout the 90s, it was discovered that vertebrates all have 3 copies of Hedgehog, not 1. So there's Sonic Hedgehog, and then they named the other 2 Desert Hedgehog and Indian Hedgehog. So we have 3 independent different genes. If you mutate any of the 3 of those, they have completely different phenotypes than each other. They are expressed in different cells. They are expressed at different times. They are different genes. Okay, so that's genes. Well, what are alleles then? An allele is an exact sequence of DNA of a gene. So this is a specific sequence for a given gene. So if, person A over there has a mutation in base 42, they have one allele. And person B over here has a mutation in base 77, they have a different allele. Now, you could call alleles mutations, but when we refer to a wild type, what we're referring to is sort of that generalized consensus that may not actually exist, but that 90% of the people have the same base at point A, point B, base 1, base 2, base 3. That's sort of considered the wild type. And the mutants or the alleles are the exact specific base sequences. Excuse me. Now, you have 2 alleles for almost every single gene you have. It could be that for any given gene, you have an identical sequence from mom to the sequence from dad. The 2 of those might be identical, so you have the same allele on both chromosomes. Well, you're homozygous in that case. You're homozygous for that allele. A lot of alleles are indistinguishable from each other. So if you have 2 indistinguishably different alleles, you're considered homozygous anyway in terms of phenotype. But your genotype is a little different. So an allele is a concrete, specific thing, whereas a gene is a specific region of DNA, but not necessarily a specific sequence of DNA. So an allele is a subset of a gene, if that makes sense. And those are the 2 that most frequently get confused. I want to make sure that I lay out the differences between them, and I want to make sure that you understand them so that you use them correctly in an exam situation where you might have some short answers you have to answer. Now, a gamete is a cell. It is a haploid cell that has one copy of each chromosome of the organism. And therefore it has one copy of every gene, and it only has one allele of each gene, the one copy. So in that case, in a gamete, a gene in a gamete is a specific individual allele. It's not sort of the broad summation of alleles that formulate a gene across a species. And then the other term that gets confused is locus and loci. Well, locus is a Latin-based word, so locus is singular, loci is plural, and they are the same word. And a locus is a physical location. It is a spot on a chromosome. So you can say, I found a mutation at this locus. Now, you don't know if that mutation is in a gene or not. It is in a specific location on a chromosome. It could be in a gene, it could be in an intron, it could be in a coding region. And I should point out that alleles don't necessarily have to be in a gene. They could just be in a region of DNA that affects how the gene works, which we're going to get into those later in the semester, either promoters or enhancers. A locus is a spot on a chromosome. Loci are multiple spots on a chromosome or multiple chromosomes. So please keep those separate in your head. Let's get started on chromosome packing, however. This is a fun topic. So as I discussed in the last lecture, the chromosomes, the DNA strands are incredibly narrow and incredibly long. They're only, about 10 to 20 angstroms wide? And each chromosome on average is almost a centimeter long, compared to 10 to the negative 10th narrower than that. It's insane. So the cells don't exist, the DNA in the cells don't exist just as nucleotides, a long polymer chain of a double helix. They do exist in the cell combined with proteins. And the number one most important of these proteins is the histones. And histones have very high histidine and arginine content. Those are amino acids with positive charges. And they bind well to the DNA, which has overall a negative charge. Specifically, the phosphate backbones are negative. So the histones bind very well to the double helix. And histones are a multi-protein complex. There's about 8 proteins, 4 different proteins, each present twice in a histone. And they form sort of almost like a yo-yo shape. And the DNA wraps twice, or just less than twice, around each histone. And what you find is when the DNA wraps this almost 2 times around the histone, you get about 200 bases worth of DNA wrapping around, ballpark. And this is uniform, this is constant, this is not variable. What is variable is how closely the histones are to each other, how close they are to each other along the DNA double helix. And that doesn't happen by the histones sliding up and down the double helix. That's more just a matter of how wound the DNA is. If it unwinds slightly, so there's only 145 bases around the histone instead of 200 bases, then there's going to be a wider spacing between them. If it winds up a little more, so there's 220, 230 bases between them around each histone, then the histones themselves are going to be closer together. So this is a great way to pack DNA more densely. And it's also a great way to protect the DNA because it's not just loose out and about risking getting torn. It's wrapped around something that's going to keep it safe. So let's have a look at what histones look like under a microscope. This is not from a light microscope. You can't see this with your own eyes. This is a photograph taken from an electron microscope. So these things are very minuscule. They're the size of angstroms, which is 10 to the negative 10th. But as you can see, this looks almost like beads on a string, is the most common analogy used for it. And each one of those beads is a histone with DNA wrapped around it. So you are actually looking at DNA and protein here. The cool thing about histones is they can change what the DNA wrapped around them does. So let me go to the next slide here. What you're looking at now is called a ribbon diagram, and it's a common and popular way to visualize proteins and nucleotides. So if you look at this, the sort of green and orange double helix around the outside, well, that's your DNA strand. That's your double helix. Inside, you've got all these sort of twists and curls. They look almost like, very tightly curled hair. Those are alpha helices. These are proteins that you see in the center. And the two images here are sort of a top view and a side view of the same thing. And this is a histone complex with a double helix wrapped around it. Now, each of those sets of coils inside the histone, each color of those is a different subunit. So the subunits are, H2A, H3, H4, H2B, and so on. Each of those is represented by a different color of curls and coils. Now, what's so fascinating about this is if you look at the far right of the slide, you can see 3 or 4 little wisps coming out. There's a blue one coming up the top, there's a green one sticking out the left side of that, and there's a yellow one down in the bottom right corner. These little wisps are the ends of the protein for each of the histones, and they're called the histone tails. Now, the amino acids in these protein tails can get modified. So I don't know how well you understand the structure of amino acids, but there's the standard core of it. That's an NH2, the amine group. There's the CO2, and there's the other carbon, and then you've got the R group, and the R group is just variable for each amino acid. Well, the R groups can be modified by other proteins. They can either have an acetate group attached to them, or a phosphate added to them, or even a methyl group added to them. Those are the 3 most common. And depending on which amino acid sticking out on these tails gets modified, and depending on how it's modified, that changes how tightly the DNA will get wrapped around the histone. It will change whether or not the histone will allow the DNA to become transcribed. It will modify all kinds of properties of the DNA. It might also modify whether or not a protein can come to bind to that section of DNA. And when we get to enhancers and repressors later in the semester, this will come into play there. But for now, I just want to point out that not only does this wrap the DNA up and condense it, it also affects how the DNA functions in the cell. So the tails of these histones get modified constantly. The methyl groups get added, the methyl groups taken off. The phosphate groups get added, the phosphate groups get taken off. Back and forth all throughout the cell, various different amino acids on each of these histone tails, some histones and not others. It's a very dynamic and complex process that we're not going to get terribly into, but you should be aware of it because it is a vital part of your cells functioning properly. Now, we've coiled the DNA twice around each of these histones, and that condensed the DNA somewhat, probably about 5 times. It's about 1/5 of its total length now, but we've still got a tremendous amount of DNA to condense. So specifically what we're talking about here is the phases of the cell cycle where the cell is undergoing either mitosis or meiosis, when the DNA needs to become condensed for it to divide. So the histones themselves sort of wrap around each other in a 7-pointed spiral. It's almost perfectly 7 histones in a loop around each other as it spirals. And this condenses the DNA significantly further. Now, we originally had the beads on a string, which was the DNA wrapped around a histone, and now the histones coiled around each other. That's called the 30 nanometer fiber. Because, believe it or not, it's 30 nanometers wide. A nanometer is 10 to the negative 9th meters. So, 10 to the negative 6 millimeters. So you can fit 1,000,000 of these side by side inside 1 millimeter. That's the width of the fiber that we're talking about here. Now, to get the sort of X-shaped sister chromatid that we are used to seeing, your double helix has to wrap around the histone, the histones have to wrap tightly with each other, and then the 30 nanometer fiber of coiled histones has to form. Then that set of coiled histones sort of doubles back across itself and makes loops. And then those loops themselves double cross back and forth against themselves to make what's called the 700 nanometer fiber. The loops themselves are 300 nanometers. And then the loops of loops are 700 nanometers. And then those coil themselves into the sister chromatids. Now, just to give you some sense of the pace of knowledge, I'm not sure when histones were first discovered or characterized, but I will say this. I took high school biology almost 30 years ago, and we had a slide almost identical to this in our textbook. The only issue is between step 4 and step 5, it was blank. And from step 5 to 7 was a giant question mark. We knew that the DNA wrapped around histones, and we knew that there was this 700 nanometer fiber that was condensed DNA that looped itself to form the sister chromatids. But that whole next to the bottom row, we didn't know any of that at the time, or at least it wasn't in the textbook, and it was just a giant question mark. So this is somewhat new information, if you consider 30 years new. But regardless, this is how your DNA compacts itself during mitosis and meiosis. But what about the rest of the cell cycle? I mean, mitosis and meiosis are a small portion of the cell cycle. So what happens during interphase? G1, S phase, G2. That's where we get into chromosome territories. Now, chromosome territories were first initially observed and proposed in the 1800s, and there was some minor work done on them from then through the 1970s. And then information about them sort of got much more prevalent and prominent from the 1970s forward. But it was not a very widely agreed upon thing. There were a lot of people, up until very recently, who considered this to be just an artifact of how people looked under a microscope, and it wasn't real. But there's very compelling evidence at this point that chromosome territories are real. And what they are is just that big loose bowl of pasta in the nucleolus during interphase. The sort of unwrapped, unbound, loose DNA that I was talking about in the previous lecture, well, it turns out that that's got some organization to it. In this diagram, each of the different colors represents a different chromosome. And what you find is, inside the nucleolus, the chromosomes kind of keep to themselves. Each one has its own little partition that it stays in, and it doesn't really intermingle with the other chromosomes. And it is actually wrapped in a sort of organized fashion within that region, which allows the proteins that are needed to transcribe genes, to duplicate DNA, to modify histones, all of these things, it allows them easy access and organized access. And it keeps the DNA from getting tangled and forming knots. And it also keeps the DNA in an organized fashion so that when it does need to condense for mitosis or meiosis, it's all right there and easily condensed. There's no aspect of the cell having to untangle a knot of extension cords or untangle a ball of yarn. It's much more organized than that. So there's not much more to this that we're going to get into. I just want to make sure that you understand that this is a thing. And this is what your DNA looks like during interphase. Let's move on. I want to talk about sex-linked traits now. I've mentioned these briefly in a previous lecture, but we're going to go into that in a little bit more detail in this lecture. Different organisms define sex or develop sex in different ways. And by sex, we actually mean gender here. In humans, gender is, through broad strokes, defined as your allotment of X and Y chromosomes. 1X, 1Y is male, 2X's is female, and then there's all sorts of anomalies besides that. As you saw in the first lab, pigeons, it's W and Z. In Drosophila, it's also X and Y. And various different organisms have various chromosomes. On a pedigree, the way to follow sex-linked traits is if the gene you are interested in, the trait that is linked to a sex chromosome, if that is on the Y chromosome in humans, it's just going to be passed down from father to son, father to son, father to son in perpetuity. There is no alteration in that. If it is an X-linked trait, then most commonly it is going to be passed through the female line and seen almost entirely in male offspring. So any males in a family that have it will be connected across generations by female blood relatives. Not through marriage, but through female bloodlines. But let's start looking at some examples. There are, in the human genome, 22 autosomes and one pair of sex chromosomes. The autosomes are the same, male or female, doesn't matter. The sex chromosomes are X and Y. Now, in humans, having a Y chromosome specifies that you are male. In insects, such as the fruit fly, that is not the case. In the fruit fly, having two X chromosomes specifies you as female. That means if you have one X, then you're male. If you have two X's, you're female. Now that's going to get a little wonky when we talk about non-disjunction in a few minutes. And also if you remember from your lab, the pigeons, the Z allele and the W allele, the ZW made female and the ZZ made male. So that's sort of the opposite here, whether you have two of the same sex chromosome or one of each of the two sex chromosomes. Well, why are we talking about this? We're talking about this because inheritance does not follow the regular Mendelian 1-to-1 pattern for sex chromosomes because you are not segregating the same number of gametes really, because the X chromosome and the Y chromosome do not have the same genes on them. So when they segregate during meiosis, it is a different pattern than the autosomes, just because one of them doesn't have the gene. So I'm going to talk to you a little bit about Thomas Hunt Morgan. He was a scientist in the late 1800s, early 1900s at Columbia University. Now, he did some really fascinating work with regeneration in planaria. He's the one that found out that you could cut a planarian into 384 pieces, and each one of those 384 pieces would grow to be a complete full planarian without dying. That was in 1902 he published that. Well, shortly after that, he decided to move on from regeneration and look at development and genetics. And instead of looking at planaria, he decided to look at the common fruit fly, Drosophila melanogaster. And he set up a lab at Columbia breeding fruit flies, and he got a bunch of postdocs and grad students, and the sheer quantity of information that these people produced over a period of 20 years is astounding. To the point where, even today, if you just say the fly lab, people who understand genetics say, oh, you're talking about Thomas Hunt Morgan in the 19-teens, 20s, and 30s at Columbia University. Several different Nobel Prizes came out of his work, and much of our current understanding of genetics also came out of his work, or the work of his postdocs. So, Drosophila, as you hopefully remember from the lab you did last week, typically have red eyes. Well, Thomas Hunt Morgan had in his lab several strains of fly that had been breeding true for generations. They were homozygous. And one day he noticed a male fly that had white eyes, just out of the blue, a mutation. And like any good geneticist, he said, oh, let's set up a cross and see if it's dominant or recessive. So he set up a cross with this male to a wild-type female. And what he found was all red-eyed offspring. Okay, sure. This is no different than Mendelian genetics. I mean, you cross a yellow pea with a green pea, or you cross a smooth pea with a wrinkled pea, all of the next generation are going to be the dominant trait. Okay, so maybe the white eye is a recessive trait. But he didn't stop there. He then mated the F1 progeny to get the F2. And what he noticed was, he got a 3-to-1 ratio, which makes sense for a recessive trait, but all of the flies exhibiting the recessive white eyes were male. That didn't make sense, because everything that was known about genetics to that point, the inheritance of traits was even in males and females, essentially. And here's one where the white eyes were only expressing in the males. Well, he could have done a chi-squared analysis on how many offspring he got and determined the likelihood that would just happen by chance, that all of the white eyes were males and instead of half male and half female. But saving himself the trouble of a chi-square analysis, he set up some reciprocal crosses. After a couple of rounds of breeding the offspring of his F2, he wound up with female white-eyed flies. And so he took one of these female white-eyed flies and he crossed it to just a wild-type red-eyed male fly. And what did he find? Any guesses? Well, this time, all of the male offspring had white eyes and all of the female offspring had red eyes. Interesting. Well, what happened when he crossed those together and got an F2 generation? Now, it was still 50-50 red and white, except it was now across gender lines. So instead of all of the females being red-eyed and all of the males being white-eyed, it was still half a red and half a white, except it was a quarter red and female, a quarter red and male, a quarter white and female, and a quarter white and male. Now we have the benefit of seeing the chromosome diagrams in front of us. Thomas Hunt Morgan did not, but he was able to deduce through these crosses that the gene that controlled color of the eyes was on the X chromosome, and the mutation that made the eyes white was recessive. And so if you have a male with the white eye allele, his eyes will be white because he only has that one allele. If he has the red allele, the wild type one, his eyes are going to be red because he only has that one copy of the gene. That gene is not found on the Y chromosome. And the females, well, they're displaying normal Mendelian patterns of dominance and recessiveness. So if you were to just look at numbers of females in all of this, then their numbers are breeding true. It's just that they have two copies, so they can have a dominant recessive homozygous, or they could have a heterozygous. The male doesn't have heterozygous. It only has the dominant, or it has the recessive. Sex-linked traits are everywhere because sex chromosomes have genes on them. This is an incredibly simplified family tree of some members of the European royal families in the 18 and 1900s. And this is a fairly famous example. Queen Victoria was a carrier for a sex-linked trait. So one of her X chromosomes was wild type, and one of her X chromosomes had a mutation for this particular gene. And this gene controls clotting of blood after injury. So when your blood doesn't clot after injury, that's a disorder known as hemophilia. Well, Victoria had many children. They're not all listed here. One of them, her son Leopold, died young, in his late 20s, early 30s, from hemophilia. 3 of her daughters were known carriers. Now, how did that happen? Well, because with her son Leopold, Prince Albert donated his Y chromosome because it's a son, and that's the only place the Y is coming from. And Queen Victoria donated a gamete that had the mutant allele. So Leopold, poor fella, he only had one copy of the gene, and the gene copy he had was defective. So he got hemophilia, and it led to his early death. 3 of her daughters, Beatrice, Alice, and Victoria, got one of their X chromosomes from Dad, Prince Albert, and that was wild type. And they got one of their X chromosomes from Mom, Queen Victoria, and that was mutant. So they were both carriers. Now, when Prince Alice had children, one of her sons died young because she was a carrier and she gave her son the mutant copy. But she also gave her daughter Alexandra, or Alex as she was known, her mutant copy of X chromosome. Now, Alex married Nicholas. Nicky and Alex were madly in love. They were also second cousins. They were also third cousins on a different family tree. And they were also both first cousins of George V, way over there on the left. So there's a lot of inbreeding going on in this family. Almost every European nation had at least one descendant of Victoria or her grandfather or father. Many of them had multiple descendants through multiple different lines. But let's step away from the inbreeding for a moment. I mean, inbreeding to the point that Nicholas II and King George, first cousins, were almost indistinguishable in person. To the point where at George's wedding, people kept coming up to Nicholas and saying, "Congratulations, how does it feel to be married?" And Nicholas was a bachelor at the time and trying to work up the nerve to propose to his cousin Alex. Now, Nicholas and Alex had 5 children, 4 boys and 4 girls and a boy, rather. You may have heard of Anastasia, if you haven't heard of any of their other children. Alex had hemophilia because he got his Y chromosome from dad, and he got his X chromosome from mom, and mom got her mutant X chromosome into him. And this actually partly led to their downfall because Alex became very close with Rasputin because Rasputin was trying to cure Alexis of his hemophilia. And he fairly convinced Alex that he could do it, and most of the population hated Rasputin. And there were many other reasons and factors leading to the Russian Revolution. But anyway, in 1918, let's just say there was no more Romanovs and move on. And these are the Romanovs. That's Nicholas II seated in the center, and Alex standing just to the right of him. And little Alexei, he's the boy off to the right. Do yourself a favor and don't ever read up the details of their murders. It's very gruesome. Now, you may remember that I mentioned another pedigree with the sex-linked trait, and this was my family, and my nephew that has Hunter syndrome, which is the mutation of a particular gene that is required for normal cellular function. In this case, the gene encodes a protein, and that protein acts as a sort of trash disassembler. Your body, in all of its normal processes and biochemical processes, makes byproducts, and these byproducts are sent to an organelle in the cell called the lysosome, and inside the lysosome there are enzymes that degrade those byproducts and break it down into parts, and then those parts are sent back to the rest of the cell to be used to build new things. Well, with Hunter syndrome, one of the enzymes that breaks down the byproducts doesn't function. And so those byproducts build up and build up and build up. And it can lead to multiple different problems. That is a sex-linked trait because the gene for that protein, that enzyme, is located on the X chromosome. Now, let's move on. We're almost done here, just a few more slides. This is an example in chickens of the barred and the non-barred alleles of wing color being sex-linked. Now, hopefully, this all looks familiar to you from the pigeonetics assignment that you were doing. And if it doesn't look familiar yet, then you still have a little bit of time to hurry up and finish that pigeonetics assignment and make this look familiar. Anyway, inheritance is wonky with sex-linked traits. Now, on to our last bit for today, and this is going to be very brief. Non-disjunction. And for non-disjunction, we are going back to the Fly Lab, back to Thomas Hunt Morgan, back to the 19-teens in Columbia. But we're not talking about Morgan. We're talking about one of his postdocs, Calvin Bridges. Now, Calvin Bridges was a heck of a character. He was James Dean before there was James Dean. He was suave. He was cool. He was a ladies' man. He had this really outrageous hairdo. And he built his own car. And he had been working in Texas for a while, and he wrote to Thomas Hunt Morgan and said, "Hey, I'd like to work in your Fly Lab." And Morgan said, "Sure." And so Calvin Bridges finished building his car and drove it across the country and had articles about himself in the newspaper in almost every major town between Texas and New York, with all kinds of beefcake photos of him standing by the car, posing in all kinds of ways with sultry bedroom eyes. This is a picture of Calvin himself at a desk that he built for himself. He was trying to draw what he saw on the slides, and the slides he's holding in his right hand are all preparations of Drosophila chromosomes. And he was trying to draw what he saw on the chromosomes. And his drawings are actually now somewhat famous for Drosophila bands. But he built this. It's basically an artist's desk, table at an angle, with a microscope attached to it. And then a little thing to cover his eyes so that light didn't get in his way. So he could sit there and stare into the microscope and then draw right in front of him while leaning back. He was also quite the womanizer and died young of syphilis. If you are interested in learning about Calvin Bridges or Thomas Hunt Morgan, there is actually a movie written about him from the point of view of his 10-year-old daughter. The movie was made in the last 15 years, I think in the last 10 years, and the 10-year-old daughter that the movie is based on was in her late 80s when she worked with the filmmaker to develop a script. The movie is called, quite obviously, The Fly Room. I have not personally seen it, but I have heard that it is amazing. So, what did Calvin Bridges do? Why am I telling you about this guy? One of the many things that he did was he noticed things. You know, there's that famous quote that the vast majority of scientific discoveries are not noted by someone saying, "Eureka, I've got it!" They're noted by someone saying, "Huh, that's weird." This is what Calvin Bridges did. He was looking at flies and looking at flies, and every once in a while, he would look at a fly and say, "Huh, that's weird." And what it turned out to be was, as he was looking at these flies, every once in a while, the white allele of the white eyes did not segregate according to the normal distribution of sex-linked genes. It was like 1 out of every 400 flies. It happened wrong. And, well, Calvin was working with Drosophila chromosomes at the time, and this is the 19-teens, 1920s. People are fairly certain that chromosomes have genes on them, but there's still some debate. There's some wiggle room there. It's not, it's not a sure thing. And so he starts looking at the chromosomes, and he realizes that in the flies where the white allele is migrating wrong, the flies don't have the right numbers of chromosomes. Some of them have an extra sex chromosome, and some of them are missing a sex chromosome. And sure enough, lo and behold, it was a perfect 1-to-1 ratio that when you had a chromosome missing, or an extra chromosome, that was what corresponded to the flies that had bizarre eye color, that it didn't match with normal sex-linked inheritance. And so he started watching these flies more intently. He developed ways to make this rare occurrence happen more frequently, and so he was able to start looking at more flies, and he was able to show that during meiosis, when the sister chromatids and the X and Y chromosome are supposed to migrate from the 2-cell stage to the 4-cell stage, where you go from double haploid to haploid, in some cases, the X chromosome, the sister chromatids did not separate, and both sister chromatids went into one cell, and the opposite cell didn't get any sister chromatids of the X gene, of the X chromosome. So for all the other chromosomes, I think Drosophila have 4. For all the other ones, everything was fine. But for the X chromosome, in one cell there were 2 copies of it, and in the other cell there were none. He also observed the same thing in some males, where in some cases the X and the Y both went into one sperm, and nothing went into the other. Now, what happens there is, in the offspring, you get the wrong numbers of chromosomes. Now, I mentioned that flies are not the same in terms of how sex is determined. In flies, having 2 X's makes you female, whether you have a Y or not. And in males, having 1 X makes you male. So if you look at the bottom right square of this Punnett square, if you have just a Y chromosome and no X chromosome, as a fly you die. If you have just an X with no Y, that's called XO, and that fly survives. It's male because it has an X, but it is sterile. It cannot produce offspring. And then the ones that have 2 X's and a Y or 3 X's, well, those are female, but the 3 X's are not all that healthy, and they usually die. So let's move on. This is going to be probably about half of the lecture because there's a lot of math in this, and it can be confusing because the math we're dealing with is statistics. And most scientists don't have a good statistics grounding. So it's confusing and tricky even to most scientists. But in reality, there's not all that much to it to be confused by, as long as you have a clear understanding of what you're looking at. So when we did the chapter on Mendelian inheritance, and we did those few lectures, we talked a lot about the 1-to-1 ratio and the 9-3-3-1 ratio of hybrid crosses, or the 1-to-1-to-1 ratio, or the 1-to-1 ratio of test crosses. Now, I mentioned in the previous lecture, the one immediately before this, that those aren't the numbers that you see, in a family of 4 or in a family of 16. You don't have exactly 9-to-3 to 3-to-1. Those are averages. And over thousands of different progeny, the numbers form those averages. They sort of coalesce around these ratios. Mendel found the 3-to-1 ratio of smooth to wrinkled peas or yellow to green peas. He found those ratios by doing 8,000 F2 peas or F1 peas, and out of the 8,000, it was almost perfectly 6,000 to 2,000, and he got a 3-to-1 ratio. But on any given seed pod that has 4 or 5 seeds in it, who knows? It's a flip of the coin for each individual seed, for each individual pea. It's a flip of the coin. And that's 50-50. And now it's not technically 50-50 here because it could be any one of 4 things and not at equal ratios. But the point is, it's not going to be exact, and it might not even be close on a very small macroscopic level. Microscopic level. It's only when you zoom out to the macroscopic level that you start to see these ratios really get clear. So, if you have a coin that you're going to flip 1,000 times, 1,000 times you would expect ballpark 500 heads, 500 tails. Well, what if you get 450 heads and 550 tails? Does that invalidate the 50-50 nature? What if you get 650 and 350? Now are you starting to deal with a bum coin? What if you have 800 versus 200? Then you're looking at 4-1 that it always comes up on heads. That's not going to happen randomly, right? That's got to be a problem. Well, how do you know? Where's the limit? Where's the cutoff between you can expect this, this amount of range from a random average coin, but anything outside of this range is probably not normal, and there's probably something weighing on one aspect. So maybe it's a magician doing a card trick, or using a trick coin, or the coin is weighted so that it always falls on one side. But how do you know? So that's where we need the statistical tools. And there's literally hundreds of these. Statisticians over the last several hundred years have been coming up with different ways to analyze numbers and large bodies of numbers. So there's many different tests. There's the student's t-test, the chi-squared analysis, binomial distribution, Pearson's coefficients, all kinds of things. And in my education in college, this was barely touched on, and you were just expected to teach yourself and to know it. And you're not going to get much better than that from me. We're going to talk about the binomial distribution, and we're going to talk about chi-squared. But things like the student's t-test, I recommend that you read about it and you try to, a lot of the places you can read about these, the explanations are very dry and confusing. So I'm going to try and make this crystal clear for you and easy to visualize. And hopefully once you have an understanding of this, going to read up on other things will be easier. That is my goal. So let's take this slowly and carefully. So a binomial distribution, if you think of, it can be called a Gaussian curve or just a standard bell curve. That is a wide spread of continuous possibilities. A binomial distribution is when you have choice A or choice B, not, choice 1 through 1,000. Binomial is flip a coin, heads or tails, have a kid, male or female. Obviously, biological gender is a bit of a spectrum depending on many different possible mutations in the determination of gender. But using simplistic examples of Mendelian genetics, you're looking at male or female, heads or tails, left or right, smooth or wrinkled, black or white. This is binary choice. And when you deal with binary choice, there's some math you can do to find out whether or not the amounts of results you're getting make sense or not. So in the textbook, in this chapter, Hartle mentions the possibility of having 3 children in a sibship. So 3 siblings together. And that there's 4 possible combinations. If you don't care which one's the oldest, which one's the youngest. You can have all girls, all boys, 2 girls and 1 boy, or 1 girl and 2 boys. And if you look here, under the 2 girls and 1 boy, there's 3 possible combinations of that. The boy could be first, the boy could be the middle child, the boy could be the youngest. But in this case, we don't care what order the boys and the girls are born in, we just care how many of them there are. So obviously, there's 8 possible combinations if you care about the order. And in this case, all girls is only 1 out of those 8 possible combinations. All boys is only 1 out of those 8 possible combinations. 2 girls and a boy? Well, that's 3 possible combinations. and one girl and two boys, that's also three possible combinations. The girl could be oldest, middle child, or youngest. Now, we learned from the multiplication rule of genetics that the probability of independent outcomes get multiplied. So, the possibility of having a girl is 1/2. The possibility of having a boy is 1/2. Now, in this case, having two girls is 1/2 times a half. We can write that as 1/2 squared. The possibility of having a boy is 1/2. So 1/2 squared times 1/2. Okay, well, that's 1/2 times 1/2 times 1/2. That's 1/8. And there are three possible combinations. Boy first, boy middle, boy last. So that's 3 out of the 8 total. So the thing that's circled that says second term there, that is 3 out of 8 possible combinations. And you can see that in the sort of organized initials there. There are 8 total possibilities, and 3 of them are 2 girls and 1 boy. There are 8 total possibilities, and only 1 of them is all girls. That's our first term. There are 8 total possibilities, and only 1 of them is all boys. That's our fourth term. So you get the point. And in this case, the total number of possibilities, we're going to add all of that up to equal 1. So 1/8 of the possibilities is all girls, 3/8 of the possibilities is 2 girls and a boy, and so on. Well, it turns out that based on the numbers of things you're comparing, there is a handy-dandy, easy way to tell what these the constants are, the terms that are circled here, 1, 3, 3, 1. And I'm hoping that you guys have had really fun calculus teachers. Okay, so I recorded about 10 more slides from here forward, and PowerPoint just erased it all. I'm starting over again from here. For some reason, it just erased all of my recordings, and I don't know why. I left off in the last slide by saying that I hope that you had fun calculus teachers, and I certainly did. Chuck Conti was my high school calculus teacher for 2 years, and he just made everything fun. And he did a lot with the Pascal's triangle, which is what we're looking at here. Pascal's triangle was discovered independently in 6 or 7 different parts of the world and throughout different times in history. And once you get the hang of it, it's really easy to do, and it turns out it shows up in all kinds of things, especially with binomial distributions. So let's talk about it. If you look at this, you see on either side of the sort of diagonal lines of ones, it's ones running down both sides. Now imagine that there's an invisible 0 on the outsides of those ones. So the first line will be 0, 1, 0. The fourth line will be 0, 1, 3, 3, 1, 0, and so on. The way Pascal's triangle works, if you're not familiar with it, is you add 2 numbers that are next to each other on a line, and their sum you put between them on the next line down. So we've got these invisible zeros. So let's look at the very top line, the one in blue that just has a 1. We've got an invisible 0 on the left. Add that to the 1, we get 1. So we put that between them 1 row down. We do the same thing on the right side. 1 plus 0 equals 1. Put it between them 1 line down. So now we've got, on the second row, we've got 2 1s. We'll do the same thing. Add the 0 and the 1, you get a 1. Add the 1 and a 1, you get a 2. Add the 1 and a 0, you get a 1. So that's our third line, 1, 2, 1. Now as you go down to the next line, 1, 3, 3, 1, I hope that you recognize that that's what we had with the 3 siblings on the previous slide. Those were our coefficients. And coefficient is the word I was trying to find there. and I just kept saying constant and other things. Coefficient is the word I was looking for. So if you look over to the left side of this graph, or image, you've got the column that has an n at the top, and that's the number of items that we're looking at in this case. So we're on the fourth row, so that's 3 items. And our 3 items are the 3 siblings. Whether that's girl, 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 boy, 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 girl, whatever. When you have 3 items to choose from, and there's 2 possible options between them, the equation you're looking at is going to have the coefficients of 1, 3, 3, 1. Now, if we had to have 5 siblings of any order and any number of boys and girls, then there will be 1 possible option of 5 girls. There will be 5 possible options of 1 boy and 4 girls, 10 options of 3 girls, 2 boys, and so on across. So this is Pascal's triangle. It's useful for a ton of other possible options, possible things, and you can do all kinds of fun math with it. We're going to leave it here for now, because we're not going to use it for anything else for our purposes and for this course. But I highly recommend that you check out YouTube videos by Vi Hart, V-I-H-A-R-T. She has a series called Doodling in Math Class that makes complex mathematics sound simple and fun, and it makes you think about math in totally different ways. I strongly recommend that you watch some of her videos just for your own education. She has since become a teacher at the Khan Academy, and one of her videos deals with Pascal's triangle. Let's move on. This is the more complex thing that we're going to deal with. I spoke a lot about this the first time I went through here, but it all got erased, so I'm going to have to do it again. First thing you need to know is factorials. That's the exclamation point that's next to the N, the S, and the T in the formula here. And I'll explain factorials a little more in the next slide. What we have here is a way to figure out the probability of having a particular outcome. So in the previous slide, we were looking at, well, the one before Pascal's triangle, 2 slides ago, we were looking at the 3 siblings. And let's say we wanted to pick one that was 2 boys and a girl. So 2 brothers and their sister. What's the probability out of all the possible sibling options that we would come up with that a family of 2 parents and 3 kids would have as their children 2 boys and a girl? The way to figure this out is this formula is 2 distinct parts. There's the fraction that has a lot of factorials in it, and then there's the p to the s and q to the t component. We're going to talk first about the factorials. Now in this case, what we're actually looking at is the number of different combinations that you can have for whatever you're looking at, whether it's 2 brothers and a sister. And that would be, sister oldest and the 2 brothers, or sister is the middle child, or sister is the youngest child. How many different combinations? So for us, we're going to go back to Mendel's peas, and we're going to think about a dihybrid cross of yellow and green peas. So we have heterozygous yellow peas that each have an allele for the green peas. And we're going to cross those. Now, we have 2 possible outcomes from this. You can either have a yellow pea or you can have a green pea. What we're looking at in this case is we're going to look at one pea pod that has 4 peas in it. And this pea pod has 3 yellow peas and 1 green pea. It's perfect Mendelian ratio, 3 to 1. But what's the likelihood, what's the probability that that's exactly what we're going to get. I mean, if you're going to look at the progeny of this dihybrid cross, or monohybrid cross rather, and you're going to look at the progeny of thousands of P's, you're going to expect it to converge towards a 3-to-1 ratio. But if you're just looking at a small handful of P's, it can be wildly different than 3-to-1. So let's figure out how different and how likely it is. So let's think of how many different ways that you can have 4 P's where 3 of them are yellow and 1 is green. Hopefully, with just a little bit of thought, you come up with the answer 4. Not because the formula is there, but because there's only 4 ways you can arrange it so that you have 1 green and 3 yellow P's. But let's walk through this math here. So on the top of the first fraction, we have the 4 exclamation point, and that means 4 factorial. And factorial is just the product of all the numbers from 1 to the number you're interested in this case 4. So that's 1 times 2 times 3 times 4. That's 4 factorial. And in this case, that means 24. Now, to find out how many combinations of 3 yellow and 1 green P you can have, you divide that 4 factorial by 3 factorial because you have 3 P's, and 1 factorial because you have 1 green P. So 3 factorial is 1 by 2 by 3, and 1 factorial is just 1. And looking at the math there, you see that that's 4. Let's show this visually. We have 4 P's. In this case, 1 of them is green, 3 of them are yellow. Well, in our first possible outcome, the first P is green. In our second possible outcome, the second P is green. But the other 3 are all yellow. I hope you see where this is going already. Our third possible outcome is that the third P is green. All the other P's are still yellow. And our fourth and final outcome is that the green P is the fourth P. So there are only 4 combinations you can have of 3 yellow and 1 green P. And that's what this fraction of factorials is telling us. Now, that formula we were looking at had the fraction of factorials, but then it also had p to the s and q to the t. I'm going to point out here that the s term is our 3 factorial, and the t term is the 1 factorial. But we're not taking the factorials of these s and t, we just need the 3 and the 1. And let's walk through why that is. Remember, we're trying to find out what is the chance of 4 P's showing this perfect 3-to-1 ratio in a 4P p-pod. So we've got there's 4 possible combinations fraction here, the 4 factorial over 3 factorial times 1 factorial. And then we add in the p to the s and the q to the t. Now in this case, the p is the likelihood that we're going to have a yellow p. And we know that it's a 3-to-1 ratio in perfect Mendelian genetics. So 3 out of 4 P's is 3/4. And we know that the likelihood of getting a green P is 1 in 4. So that's 1/4. Now, we are saying that we want 3 P's that are yellow. Each yellow P has a 3 out of 4 chance. So the first P has a 3 out of 4 chance of being yellow. The second P has a 3 out of 4 chance of being yellow. And the third P has a 3 out of 4 chance of being yellow. So that's 3/4 times 3/4 times 3/4. Because remember, this is the multiplication rule of genetics. Each of these P's are a completely independent mating of gametes. So whether it is homozygous yellow, heterozygous yellow, doesn't matter. There's a 3 out of 4 chance of it being yellow for each P, and there's 3 of them. So we have to multiply 3 out of 4 times 3 out of 4 times 3 out of 4. And so that's why we have a 3/4 cubed there. So the S is that 3, the cubed. Now we're looking for 1 green P. Well, there's a 1 in 4 shot of a p being green, and we only want one of them, so it's 1/4 raised to the 1. And again, that 1 is our 1 factorial, it's the t. So when we multiply all of this out, we wind up with 27 over 64, which is about 0.41 or 41%. What does that 41% mean? It means that if I show you a pea pod with 4 peas in it, 41% of the time from a F1 monohybrid cross, 41% of the time you would have that perfect 3-to-1 ratio. Sometimes you'll have a 2-to-2, sometimes you'll have 4 yellows and no green. Once in a while, once out of every 256 times, you'll have all green. Can you figure out how I did that math? So that is how we use this formula. It's telling us the likelihood of having that 3-to-1 ratio, the probability of having it. Now, I want to circle back to the fractal fractions, because what we looked at there was 4 possible options. But I want to show you what that looks like if we get a little bit more complicated. So this is a field of mathematics called combinatorics, and it's not my strong suit, but it's fun. So let's say we want to find out if I open up a pea pod and there's 2 green peas and there's 2 yellow peas in it, what was the likelihood of that happening out of an F1 cross? Well, let's find out. In this case, there's 4 peas, so the numerator of the fraction has to be 4 factorial. Now, we're looking at 2 yellow peas, so that's 2 factorial in the denominator, and 2 green peas, so that's another 2 factorial in the denominator. Well, if you do the math of all the factorials, the answer is 6. And if you look at the diagram, well, there are 6 possible combinations of 2 and 2 peas. Okay, sure. Now, what's the probability that you're going to get 2 and 2 peas? Well, then it's going to be that 6 times 3/4 squared, because each yellow pea is a 3/4 chance of happening, times 1/4 squared. And that winds up being 6 times 9 over 64, which is, let's do the math in our heads, 6 times 9 is 54 over 64. So the chance of getting a 2 out of 2 combination, that seems pretty high. Check my math. Did I do that right? There's a strong likelihood I made a mistake there. Well, what about 5 peas? Let's look at 5 peas where 2 are green and 3 are yellow. Using that same fraction, it tells us that we have 10 possible scenarios where we can have 2 green and 3 yellow peas. And then if you look at the diagrams, there's all 10 of them. So this is all looking at the binomial distribution, because binomial in this case because there are 2 possible options, green or yellow, boy or girl. But let's move on to something a little bit more complicated, and we're going to check out the chi-squared analysis next. That's going to be next, but for right now, this lecture has gotten pretty long, so as I suspected, we're going to move chi-squared analysis into lecture 6. So, see you there.